we've talked about several different mechanisms or modes of inheritance. We've talked about autosomal dominant and autosomal recessive, and an example of that would be Gregor Mendel's garden peas with the purple color being dominant to the white color. We have talked about multiple alleles and a few examples of those. One example that we did discuss was human hair color. It is not just one gene with two different possible alleles, but in fact there are multiple alleles that are involved that do in fact result in all of the different colorations that we do see in terms of human hair color. We have talked about incomplete dominance. In this case, we don't have one allele that is dominant over the other one, but in fact what we have is a blending together. So an analogy that we used was taking paint and mixing paint together. If you have a can of red paint and you have a can of white paint and you mix them together, what you get is the blended or intermediate form, which would be pink, and that blended form is an example of incomplete dominance. Codominance is another one that we discussed. This is similar in some ways to incomplete dominance in that one allele is not dominant over the other, but now in the heterozygous form, it is not an intermediate blending, but we see both of the phenotypes from the homozygous forms that are visible and present in the heterozygote. To go with the paint example again, it would be like taking red paint and white paint, mixing them together, and instead of them mixing and forming pink, you see some clumps of red and you see some clumps of white. And we've also talked about two-trait crosses. Along with two-trait crosses, polygenic traits, traits that are controlled not by one single gene but by multiple genes. And an example of that would be something like human eye color. And we've also discussed briefly epistatic interactions. We have one more mechanism of inheritance to go, and that is the point of this presentation. So as we talked about, Gregor Mendel did his work in the middle part of the 19th century. He presented his findings and no one was really all that impressed at the time. And as you can see, it sort of sat for about 35 years. But over this period of time, new staining techniques in microscopy allowed them to observe the chromosomes. And they could see that these chromosomes inside of the nucleus, they in fact begin to separate during cell division, mitosis and meiosis, and they began to piece together the events of cell division. And then there was the realization that these traits that are passed on from one generation to the next, the information for those traits in fact is found on the chromosomes and genes that are contained within the chromosomes. And the two gentlemen that were responsible for figuring this out, an American, Walter Sutton, and a German, Theodor Boveri, both figured it out independently at about the same time, and that is now referred to as the Sutton-Boveri chromosome theory of inheritance. We see another cross here in some ways similar to what we began to talk about with Gregor Mendel's garden peas. Here we're talking about the wing color in moths, and we have our original parental generation, light-winged females and dark-winged males, and we can see that in the F1 generation, all of them have the light wing color. So based on what we have talked about, we only see one of the phenotypes in the F1 generation. We would then make the assumption, and we're going to see that in fact this will be incorrect, but we would, based on this alone, make the assumption that light is dominant. So we will use a capital L to represent the light wing color and a lowercase l to represent the dark wing color. Using these letters, we can then write down the genotypes for each one of the parents. That would make the females homozygous dominant and the males homozygous recessive, and all of the offspring, because they get one allele from each parent, all of them would have a capital L and a lowercase l. Now you might recall that Gregor Mendel did something very similar to this with his garden peas, but then what he also did was the reverse cross. So with the reverse cross, in this example, what we have are females that have light wings, males that have the dark wings. With the reverse cross, we're just flipping that around. So now what we're going to have is the females that have the dark wings. So using the same symbols that we used before, we would say that the female has two lowercase l's, the male would have two capital L's. But if we take a look at the F1 generation, we see something here that we've never seen before. We see for the first time a difference between males and females. So what we can see is that all of the females in the F1 generation had one phenotype, and all of the males in the F1 generation had a completely different phenotype. Now we'll come back to this shortly, but let's jump ahead and take a look at some of the work of another fairly famous biologist at the time, Thomas Hunt Morgan. He was taking a look at the most studied species in genetics, and that is the fruit fly Drosophila melanogaster. So we can see a picture of the Drosophila melanogaster at the bottom here. This is a typical fruit fly that you would see in the wild, and for that reason it is called wild type, and it has certain characteristics, and we can see here that one of those characteristics is the red eye color. Now, just like Gregor Mendel chose the garden pea for specific reasons, Thomas Hunt Morgan was very fortunate in choosing the Drosophila, and he did choose it for specific reasons. With the Drosophila, there is a very short life cycle. So whereas Gregor Mendel could grow 2 or 3 generations in a single year, well, Thomas Hunt Morgan could grow many, many more generations for the Drosophila because they have a life cycle of only 12 days. Gregor Mendel had hundreds of offspring after he performed his crosses, and it's the same story with the Drosophila. There are hundreds of eggs, and so there is a very large sample size that can be obtained. With Gregor Mendel, easily identifiable characteristics in the garden peas. The flowers were purple or they were white. The seeds were green or they were wrinkled. They were yellow or they were green. And it's kind of similar with the Drosophila. You do need to take a look at them underneath the microscope, but it's very easy to see, for example, the eye color. So the eye color may be red, it may be purple, it may be white, there may be what are called bar eyes, or there may be no eyes. Similar with the wings, we can see all of these different wing shapes, so they are easily identifiable traits. In addition to that, compared to humans that have 23 pairs of chromosomes, Drosophila only have 4 pairs of chromosomes, or 8 chromosomes in total. This made it a lot easier to observe the chromosomes, to follow the chromosomes, and to take a look at the traits that we find in Drosophila. Now we have talked about this before, but Morgan found that in females there are 4 homologous pairs of chromosomes. In other words, they all pair up. All 8 of them, they arrange themselves into pairs. But in males, there are only 3 homologous pairs of chromosomes, and the last one isn't really a pair, they are non-identical. Now if we take a look at the picture here that is showing the male chromosomes and the female chromosomes, we can see that in the case of the female, this one is a pair, this one is a pair, this one is a pair. Those are all homologous chromosomes, and so is this last pair, homologous chromosomes, and those are both the X chromosomes. If we take a look at the male, we can see that we do have 3 pairs of homologous chromosomes, but if we take a look at these ones, we can see visually that they are not the same. So even though they pair up side by side, even though they separate during mitosis and meiosis, they are in fact different. So in the case of females, they have 2 chromosomes that are very similar. They have the similar information, but in the case of males, there is completely different information that is found on the Y chromosome. So yes, in the case of the females for fruit flies and for humans, there are 2 X chromosomes. And we see that the male fruit fly, just like in human males, has 1X and 1Y chromosome. And keep in mind as well that information that you find on the X and the Y chromosome, it is not the same information. It is completely different information, completely different genes. When we do discover that we do have a trait that is on the X or in fact on the Y chromosome as well, you do need to use some different notation. You need to make it very, very clear that that particular gene and the versions of that gene, the different alleles, are on the X chromosome. So what we use is a capital X to represent the X chromosome, and then you superscript your other letters whether they're dominant or recessive on the X chromosome. So in this example here, we're taking a look at the eye color in Drosophila. The normal wild type eye color is the red eyes, and it in fact is dominant. So I'm using the symbol a big X and a superscript capital R. The white eyes is recessive, and so I'm using the same capital X, and this time I'm putting a lowercase r on that X chromosome. We will find with this example they do use some slightly different notation. What they use is a W with a plus for the wild type, and whenever you do see the plus, that does simply mean wild type when we're talking about the Drosophila. And they use a lowercase w without the plus to represent the white eyes. So if we take a look at this cross that you have, what we can see is that we have two parents. We have a female, and again, this is the symbol here for a female that has red eyes. The female has two X chromosomes. The male has, of course, an X and a Y chromosome. So what we should have on the X chromosome for the female, if this is a pure breeding female, is a capital R on each one of those X's, and for the male, it would be a lowercase r on the male's single X chromosome. If we do the cross here, I won't show the Punnett square at this point, but from the female, all we have are capital R's. So all of the offspring, regardless of whether they're male or female, will have a capital R. From the male, if it is a female that is produced in the F1 generation, she will get the X chromosome from the male, and the X chromosome from the male, of course, has the lowercase r. If it is a male in the F1 generation, that male will get the Y chromosome. So these are the two possible genotypes in the F1 generation. And as we can see, both of them do have a capital R, so they will all have red eyes in the F1 generation. Now, what if we take these two offspring from the F1 generation, and they now become the parents for the next generation? So now we're crossing these two genotypes here. When we cross these two, we will see a Punnett square a little bit lower down, but just kind of thinking our way through this, we can take the capital R from the female, the capital R from the male, and what we will find is that a number of the females will then be homozygous dominant for this particular gene. We can take the capital R from the male and the lowercase r, X lowercase r, from the female. These female flies will have the same phenotype. They will have red eyes as well because they do have at least one copy of the dominant allele. For the males in the next generation, we would have to have the Y chromosome that's coming from the male, so we can have either the normal dominant allele from the female that would produce males that have the red eyes, or we would have the mutant white version coming from the mother, the Y chromosome coming from the father, and now we see here that we have a difference between the males and the females. So this is the key that you're really looking for, is a difference between the males and females. Now we can just go down and take a look at the Punnett square that you do have on your sheet. And here again, they're using notation that is slightly different than notation that I was using, but they're showing really exactly the same thing, what we see in the F1 generation, and a little bit further down, what you see in the next generation in the F2 generation. Now, when you are writing down your results for these offspring, it doesn't always work great to write down a ratio. Sometimes it's better to write down a fraction. So in this case here, what we could say is that all of the females, even though they do have a different genotype, we can say that all of the females do have the red eyes in the F2 generation. Now we do need to specify that it's different for the males, and it needs to be very clear that there is a difference. So what we have is that 1/2 of the males are going to have red eyes, but 1/2 of the males are going to have the white eyes. Let's go back up to our original example where we were taking a look at the wing color in moss, and now we can see that we have a little bit of a problem because if we take a look at the reverse cross, there's a difference between the males and the females. As soon as there is a difference between the males and the females, whether you notice it in the F1 generation, the F2 generation, the first cross, or the reverse cross, that is an indication that it is what we call a sex-linked trait. Most of the time we're talking about these traits being on the X chromosome. If it is a trait on the Y chromosome, then only males will ever have it because only males have the Y chromosome. So because we see a difference here between males and females, now we need to use the different notation. So we can stick with the letters that we have, a capital L for light, a lowercase l for dark. However, I need to put those on the X chromosome because these are X-linked traits. So now we're using X with a big L for light, X with a little l for dark. Females do have two X chromosomes. So the light-winged females would be X big L, X big L. The males would have one copy of an X chromosome with the lowercase l, but now we need to get rid of the second L because the males don't have another X chromosome. They, of course, have a Y chromosome. So in the F1 generation, the females would have this genotype, heterozygous. The males, however, are going to end up with X with big L because the X chromosome in the males always comes from the mother, and the mother only has X big L. The Y chromosome comes from the father, and that's why we see that in the F1 generation, all of the offspring for this cross will in fact have the light wing color. If we go on to the next one here, onto the reverse cross, again, we can make this female here homozygous recessive on the X chromosome. The light-winged males will have X with the big L, but again, let's get rid of this second L and stick in the Y chromosome. So now what we can see is that the females in the F1 generation, they have to get an X chromosome from mom. Mom only has the X with the little l. And from the father, they have to get the X with the big L. I'm just going to write the X with the big L first, and then the second one from the mother, X with lowercase l. In the case of the male, they have to get the X from mom, that's X little l. They have to get the Y chromosome, and that is why all of the males in this cross will have the dark wing color. So now let's talk a little bit about humans. In humans, of course, it's more complicated in terms of the chromosome number. There are 23 pairs of chromosomes, or 46 chromosomes in total. The first 22 pairs of chromosomes, you might recall, are the autosomal chromosomes. And the remaining pair, which is really only a true homologous pair in females, are the sex chromosomes. And again, females, they have two X chromosomes, but in males, it's not exactly a homologous pair. They have an X and a Y chromosome that don't only have different versions of genes, they have completely different genes. Both male and female sex chromosomes, they do pair up and they do separate during meiosis, but there is no crossing over that occurs. At least that is the case in males. There is no crossing over that occurs between the X and the Y chromosome. In females, there is crossing over between the two X chromosomes. And a couple of kind of important points and useful to remember: fathers pass X-linked alleles to all of their daughters. Males only have one X chromosome. All females, all daughters, will get that X chromosome from the fathers. And a female will only express an X-linked recessive allele if she is homozygous, and we'll see a couple of nice examples of this. But males, they only need one recessive allele to express it. And that is a really important point, and that is the reason why when we are talking about X-linked recessive traits, they are much, much more common in males than they are in females. We'll take a look at a couple of different traits here on the X chromosome, and I've also thrown in a couple on the Y chromosome. But two common ones that we do talk about for traits on the X chromosome are for blood clotting and for color vision. So the normal version for blood clotting is dominant. So that would be X with the big N. a big N, that is for normal blood clotting. So if you do cut yourself and you're bleeding, whether internally or externally, you need to be able to seal off those blood vessels. And someone that does not have what we call a normal blood clotting factor is unable to seal off those blood vessels. It can be for various different reasons, and there are several different kinds of hemophilia, but hemophilia does result in the inability to seal off those blood vessels. So in its severe form, people do bleed to death unless there is medical intervention. The other one that I have here is also X with a lowercase n, but now it is lowercase n prime. This one is for color blindness. The normal version, again, is dominant, so I'll put X big N with a prime, and that is for normal, in this case, color vision. And as I mentioned, for X-linked recessive conditions, and these are X-linked recessive conditions, what we will find is that both hemophilia and color blindness are much more common in males than they are in females. The two that I have here on the Y chromosome, again, these should only be traits, should only be genes that males have, because only males should have these genes that are on the Y chromosome. One of them is for the sex determining factor or sex determining region. This is the one that determines during embryonic and fetal development whether it is the testes will form. If there is the Y chromosome and there is this gene, then the testes will form. And the other one is for something called azoospermia, which is the inability to produce a sufficient number of sperm. Here I'm just showing the female X chromosomes, and I'm showing on this first chromosome here the two different traits for blood clotting and for color vision. And we can see the gametes that would be produced if we have this first chromosome here. We would have one gamete that has the defective version for blood clotting, but the normal version for color vision. And on the other chromosome that will end up in the other half of the gametes, we will have the X chromosome that has the normal allele for blood clotting and the defective allele for color vision. For the male with only the one single X chromosome, we can see that what that male will contribute on the X chromosome is defective versions of both the allele for blood clotting and also for color vision. And just a reminder again, why do I not have these N's on the Y chromosome? Because there is no information on the Y chromosome for either one of these traits. One other point, and kind of an interesting and important point for you to know about the X chromosome, is something referred to as X chromosome inactivation. In every female, in every single cell of every female, there are, of course, two X chromosomes, but there is only one of them that's actually contributing to the phenotype of that particular cell. And the reason for that is that out of the two chromosomes that a female has in her cell, one of them is inactivated. When it is inactivated, it becomes condensed, it becomes highly folded up, and when it is condensed and highly folded, the information on that chromosome is not accessible. It is not what we call expressed. That information cannot be read, and it forms a very condensed structure that you see under the microscope that is referred to as a bar body, and you do need to know that term. The bar body is the inactivated chromosome that you find in all of the cells of every female cell. So the chromosome that is inactivated is entirely random, which means that out of all of these cells in a female's body, approximately half of them will have one chromosome, one X chromosome that is active, and in the other half of the cells, it will be the other X chromosome that will become activated. So if we take a look at this picture that we see here, it's showing that in the original zygote, this is the X chromosome that came from the father, the paternal chromosome. This is the X chromosome that came from the mother, the maternal chromosome. And this picture down at the bottom here is just a representation of an adult female. and all of the cells in that adult female, which again, if we're talking about a human, could be trillions of cells, and it's just showing that we have this sort of random distribution where some of the cells have one X chromosome turned on and other cells have the other X chromosome turned on. This does lead to some interesting situations, and this is definitely an interesting one here. So we're talking about cats in this example, and what this is showing in this picture down here is what is called a calico cat or a tortoiseshell cat. So it does have a different coloration pattern. What it does have is some black fur, and it has some orange fur. And the reason for the different color of the fur is that there is a different gene that is used, a different gene that is expressed in the skin of these cells that is producing the hair. Now this fur color is a trait, is a gene that is on the X chromosome. Now, because it's on the X chromosome, one of the two X chromosomes is going to be inactivated. So if you have a female cat, which is heterozygous, they have one copy of the black allele on one chromosome and the orange allele on the other one, it is going to be random in terms of which gene forms the bar body and which one is active. So we can see that in the hair that is producing the black fur, we do have the active X chromosome, which has the information for the black pigmentation. The other one is highly condensed, and that is what is forming the bar body, and that one is not being used. In the cells that have the orange or red coloration in the cat, it's the opposite. It is the other chromosome that is being used, the information is being read, and that results in the orange pigmentation, and in this case, it is the chromosome that has the information for black color, which is forming the bar body. Back to the human example, and here we'll take a look at hemophilia. This one here is what is described as a sex-linked, or more specifically, X-linked recessive condition. So it is on the X chromosome, it is recessive, and typically, of course, that means that for a recessive condition, you would need to have two defective copies in order to express that phenotype. Well, that is the case in females, but as we'll see, that is not the case in males. So if we take a look at females, remember that they do have two X chromosomes. Males, of course, have an X and a Y chromosome. In the case of the females, then there are three different possible genotypes. We can have on both of the X chromosomes the lowercase mutated or defective version of this allele, and that is the only situation where the female will end up having hemophilia. So she is homozygous recessive for this X-linked trait. If we have one of each, one normal version and one defective version, this female does not have hemophilia because she has a normal version, but she is a carrier. She herself does not have it, but she can pass it on to her children. And the last possibility is a female that is homozygous dominant and she is completely normal. In the case of the males, there is no such thing as a male carrier because males do not have two X chromosomes. They either have the normal version, they don't have hemophilia, or they have the defective version and they do have hemophilia. And again, this is the reason why conditions like this, X-linked recessive conditions, are much more common in males than they are in females, because whereas females need two copies of the defective version, males only need one defective version in order to have hemophilia. What is the explanation for this? Well, in order to clot the blood, you need to make, well, several different blood clotting factors, and there are several different kinds of hemophilia. But in order to make this blood clotting factor, you need to have a starting material called the substrate, and then you need to convert it into the blood clotting factor. In order to do that, you need some instructions, and those instructions to make the enzyme that performs this conversion come from the dominant version of this gene, which is X with big M. If you don't have that version, if it's not present, well, that means you don't make this enzyme. If you don't make this enzyme, you cannot perform this conversion, you don't have the blood clotting factor, and then that individual will have hemophilia. We'll finish this off by taking a look at three different crosses here. And these three different crosses are for hemophilia. Let me just point out the shading that is being used here. So for this shading, we have the clear boxes. Those clear boxes are indicating that that individual is normal. If it is the light shading, that is going to indicate a carrier. And if it is the dark shading, that means that they have hemophilia. So for this first cross that we have here, we have a female, no shading, so this female is not a carrier. This female is completely normal, homozygous dominant, and we have a male that does have hemophilia. One defective version is all they need, X with a little n, the Y chromosome. That male has hemophilia. Here what they have are the gametes from the female along the side. So we can see that the female, well, can only really produce one type of gamete, X with the big N. I'll write it down twice so. And for the sperm, X with a little n will be in half of the sperm, and the Y chromosome will be in the other half. So if I go on and fill in the Punnett square, we get X big, X little, X big, X little for the females. So we can say that all of the females are going to be carriers in this case. And again, this is where you need to be really specific and break it down, males and females. For the males, X with the big from mom, Y chromosome from dad, and that is the case with all of the males. So we can say that all of the males from this first cross will in fact be normal. So none of the children will have hemophilia in this case. For the second cross, what we have now is a female that is a carrier. So she does not have hemophilia because she has a normal version. And we have a male that does not have hemophilia. Female gametes, now we can have two different possibilities, X with a big N, X with a little n, and from the male, X with a little n and the Y chromosome. For our Punnett square, female here, which, I should actually have a capital N for the gamete from the male, the X chromosome from the male. So here we will have X big N for this female. Next one, we will have X big, X little. So in terms of the females, we will have one half of the females that are normal. They do not have hemophilia. We'll have one half of the females that are carriers. For the males, first one here at the top, we have X with the big N and a Y. And the last one, a male with hemophilia, X little n and Y. So we have one half of the males that are going to be normal, non-hemophiliac, and we have one half of the males which will have hemophilia. Our final Punnett square that we have here. So is it possible for a female to have hemophilia? Well, it is, but like I said, it's going to be much rarer than males that have hemophilia. In this cross, we're starting with a female that does not have hemophilia, but she is a carrier, and we're starting with a male, the father, that does have hemophilia. Gametes from mom, X big N, X little n, from dad, X little n, Y. So in our Punnett square, we end up with a female that is a carrier. And for the first time, we end up with a female that does have hemophilia. For the males, we have a normal male, and we have a male that has hemophilia as well. So how would we write this one? We would say one half of the females are carriers. One half of the females have hemophilia. For the males, one half of the males are normal, and finally one half of the males will also have hemophilia.